a couple years ago to beautiful Boulder, Colorado, where I was actually a graduate student. So although there's about a 20-year gap, it's been great to be back. And as Becky said, yeah, Matt, who's I guess his official title is a research associate, will give a great overview of making schematics and videos and models for publication purposes. He's really changed, I think, how our lab presents our work. So hopefully you'll enjoy that. So yeah, so I'm excited to give the last lecture, I think, of the week for all of you. This will build a little bit from what Bill talked about with some of the material systems. I'd say mine's probably going to be a little more fundamental of just the basics of materials, how we design them for mechanobiology. Maybe what you'll take away is some reasons you might choose one type of material, and then how we process them with a lot of different technologies. I'll give you examples towards the second half of the presentation on where these have been implicated into mechanobiology questions, but a lot of the early stuff will be kind of overview. And please do stop me if you have questions, and then I'll definitely ask at the end if you have questions as well, and maybe I'll pause in a few spots. Great, so yeah, integrating biology and mechanics through materials, and we would typically call these mostly biomaterials. So the objectives for this lecture is to talk about why you might want to use different types of materials in mechanobiology, and then how you can implement them for these different applications. I'll go through a little bit the motivation again. I think it builds off lectures you've already heard throughout the week. Again, go through just a little bit of basic material science. I've actually pulled some of that over the years in this lecture, so it's not too much about just the basics, but try to bring out important points and properties of these materials that you might be interested in. I'll definitely highlight hydrogels. That's 95% of what our lab works with are swollen hydrogel materials, and some of those properties, I think they're very useful in these applications. And we'll go through a little bit about how cells interact with these materials as 2D planar systems, and then if they're surrounded by that material in 3D. And then we'll go through some techniques like 3D printing and electrospinning that can process these materials then into the appropriate configurations for these questions. Yes, please ask questions throughout. So I think really as we've evolved, and my lab being at the University of Pennsylvania for many years, there's just a lot of great knowledge and expertise in the extracellular matrix. And so when we think about designing materials, often we look to what surrounds cells. So these are mammalian cells in their in vivo or in situ context. So if we understand what they're surrounded by in biology and in the body and in tissues and during development, maybe we can then take out the right kind of signals and cues from the extracellular matrix and implement them then into our material design. So this is probably very much overview, but if I'm thinking about a cell down here, and this is the cell membrane, it's interacting then with all these different molecules, things like proteins, large fibrillar proteins like collagen, different types of sugars, through things like receptors where it can directly bind to sequences in those proteins, or different types of receptors that can bind to then some of these sugars. And then lots of things that happen indirectly. Growth factors might be embedded or bound to the extracellular matrix, and then those can also bind then to receptors for those different factors. So how can we control then our materials to mimic maybe some of these features? ECM was once thought to be fairly inert, just kind of holding together your cells into different structures, and thought to only really kind of stabilize that structure. I think, we learn more and more about the signals that come from the extracellular matrix and how those then play a very active role in cell signaling and their behavior. And that's particularly important in mechanobiology. Usually if a cell is not interacting through some interactions like an integrins, they're not going to receive those signals. But if they can, it's a very powerful kind of signal to cells and their environment. There's, of course, ECMs are very different across the body, so we wouldn't say there's one molecule that's important for all cells in their signaling, because cells of different types and with different questions might have different types of ECM, different levels of ECM. And importantly, too, it's also a dynamic process. So especially in disease, You can see changes in the extracellular matrix. And so here, we and others are really interested in dynamic materials where their properties can change over time with some sort of signal. So that's a bit of our inspiration. If you look at some of the details, again, there's two main classes that I've already mentioned, the fibrous proteins like collagens that provide a lot of the structure and adhesion, and then the sugars like polysaccharides that often are kind of that swollen component to the ECM. So a lot of kind of diffusion of different molecules, and also things like compressive properties as they can resist compression, often due to the charges that are present on the sugars. So there's lots of, again, different structures. It's very diverse and heterogeneous. And here's just a few examples. If we just look at stromal tissues where cells are surrounded by extracellular matrix, versus something like tendons and ligaments, where there's organization. So in this EM image, you can see a lot of alignment of the extracellular matrix, or the basement membrane, where cells are just sitting kind of atop it and interacting with it on one side. All of these have different structures, different kind of signatures for the cells, different types of proteins and sugars that are present. So we might look again towards that environment in vivo. We've been interested in, and I won't talk too much about this, also in developmental signals. How does the temporal change of ECM signals with time during development might guide then things like stem cell differentiation, if we can then match or mimic some of those features. So I think lots of inspiration for material design. And as I mentioned, ECM changes in disease. And so this is from a review that Matt Davidson had put together. And this is really, Becky is the senior author on this. But we put together kind of the idea of, again, how materials can mimic features and how that changes with disease. So imagine here, these are different epithelial cells sitting on a basement membrane on top of an ECM that might have these stromal cells within it. Maybe there's some sort of injury, and we're thinking about things like that will lead to tissue fibrosis, but there's some insult that then changes maybe those cells that can then signal to these then stromal cells to then alter their behavior, maybe differentiate into myofibroblasts, deposit a lot more ECM or increase the level of cross-linking of that ECM, and then undergo this fibrotic response. And in some tissues, this can resolve. So again, you might see a stiffening during this fibrosis and then a softening behavior in some cases when that changes. So that's where maybe some of the dynamic materials are of interest in mechanobiology. And we worked quite a bit over the years with Becky on these types of platforms. Okay, so where do biomaterials really play a role? So we can replicate a lot of these signals that I've mentioned in the ECM. So things like adhesion, by incorporating different factors or biochemical signals into our biomaterials, maybe they can bind to then integrins through different ligands that we've introduced. Things like mechanics are really important. So again, mechanobiology, this is really crucial, but it ties into adhesion. The degradation of the material, not only a change in the dynamics of material properties, but in a lot of applications in the clinic, you want the material to degrade away. Also things like topography. This can be a really powerful signal to cells to organize those cells. So I'll go through some of these, and then other things that we think are important. So again, on a basement membrane, maybe that's more of a two-dimensional interaction with a material, but in lots of connective tissues like cartilage, the cells are really surrounded by that tissue. So the context at which we present materials is important. There's structure in 3D, so that's where some of the 3D printing will come up a little bit later, and we can try to, again, mimic some features in the ECM, and then also their dynamic behavior. I kind of break down biomaterials into a few different classes, and some sit at the intersections of these materials, but one of our favorite ones, as I mentioned, are hydrogels. So imagine something like your contact lens. So this is a soft, swollen material. Typically, hydrogels generally have a nanoporous porosity or structure to it, so just a molecular porosity. But then we can process hydrogels into macroporous structures. So there's actually pores on the size scale of cells by making them into things like foams and fibers. So I'll mention methods in which you can do this, but foams are really porous, spongy materials where if you want lots of cells to interact with it, you might make it highly porous. And then fibers, depending on the scale of the fibers, whether they're smaller or bigger than cells, but again, mimicking features of maybe the fibrillar structure of the extracellular matrix. So I don't think this encompasses all the materials. And of course, there's composites and mixing together of these different ones. And we can make these foams and fibers out of things like elastomers or other types of crystalline types of polymers. But I think this encompasses a lot of the ways we might design a material. Okay, so as we think about incorporating them, I just thought I'd give this one example where you can imagine maybe a cell surrounded by a hydrogel, and so embedded within that material. So how do we incorporate things like the presentation of growth factors? So in this case, we might either put in small vehicles like nanoparticles or microparticles that can release growth factors to those cells. So that's a very relevant way, I think, to do this. And if you want high concentrations of those molecules, you can definitely do that. You can add just the growth factors throughout maybe a hydrogel network. Some of them will diffuse out really quickly. So we can introduce then various signals. So one might be heparin that can then bind to those growth factors if it's a known heparin binder to then slow its passive diffusion from these materials. So that's a way we can engineer then in how growth factors are present. And you could do this with multiple factors if you have some cascade of molecules that you want to introduce to the cells. Things like cell adhesion. So I mentioned a little bit on adhesive ligands. So there's maybe peptide sequences or full proteins that you could put throughout your material that cells can bind to through integrins. You can put other molecules, just like cell surfaces are present things like CD44. You could put those into materials to bind different factors, or you could build them out of things that will bind to cell receptors like CD44. So one of those is hyaluronic acid that is a material we've worked a lot with in our lab. So again, something that's found in many tissues. Things like mechanics, we can change things like the cross-link density of these hydrogels, but there's, I think, a lot of types of mechanics that I'll mention are important. Degradation, again, I'll go through this a little bit more, but whether it's a material that just slowly degrades away in the presence of water, or whether you introduce maybe a mechanism where cells can then, maybe cell surface enzymes like matrix metalloproteases can break down that material. So either a passive or a cell-mediated degradation. And then lastly, topography. So again, I'll keep saying a lot of these different terms, topography being, again, the structure of the material at a scale that the cell can actually sense that structure. And so alignments to get, anisotropy to cells can be really important through topography. Okay, and I think this might be one of my last kind of intro slides, but thinking about the different types of properties that I've been mentioning and how cells respond to them. So for many years, we focused on the stiffness of a material and cells on soft materials because that material is so soft as they try to pull on it, they can't really generate the tension or the traction needed to spread. These cells could remain fairly rounded. Whereas on a stiff material, like a tissue culture plate, these are very rigid, cells can spread very easily. So again, you can model different aspects of diseases through then changing the stiffness and maybe mimic what's known about those tissues. I mentioned dimensionality. So again, a 2D surface versus embedded in 3D. And then sometimes we call it 2.5D. So something that has a highly topographical kind of interface with those cells. So it's not a planar surface, but it's got some features to it. There's been a lot of interest in things like viscoelasticity. So other types of mechanics that are found in materials. So if you have more of this viscous rather than just purely elastic component, that can alter how a cell senses the material properties when it pulls on it. Things like deformability. So on a fibrous material, if they can pull all these fibers together, can change how a cell responds. Plasticity, so whether a cell can actually change that material so it has a plastic response. So maybe it's moving through that material or again, pulling on fibrous ones, but as it interacts with it, it's changing that material properties. So this is bidirectional. And then porosity, and I heard porosity as a question. The scale of porosity is really important. So again, a lot of materials have a nanoscale porosity that a cell can't migrate through. So they'd have to degrade a material to make their way through it. But that might be enough to get diffusion of different molecules through the material. Whereas a microporosity, a cell might be able to just find its way through a path, whether it's incorporating degradation or not into that. So lots of things. And I think as you read papers on mechanobiology, a lot of these terms will come up. And so thinking about how the authors what they mean by those properties and what they're really exploring, I think can be important. Maybe one comment too is that a lot of these are really coupled together. So it might be hard to change only stiffness in a material because that's also going to change something like degradation. But each of these have been shown to influence cell behaviors. Okay, so that's a lot of the motivation and I think some of the general properties of materials that might be important for mechanobiology. So I'm just going to spend a couple slides on thinking about some of the fundamentals of the material science and how we test these types of properties that are of interest. So as I heard Bill had said, and also kind of thinking about synthetic materials versus natural materials, both of these I think are really relevant, and then call it hybrid materials that kind of sit somewhere in between those. So synthetic materials, he mentioned polyethylene glycol. This is one that's very popular, but also things like polyacrylamide. So something that maybe you've used for protein gels is probably the most widely explored for two-dimensional studies of mechanobiology because it's very easy to change the material properties of a polyacrylamide gel. Natural materials get much more complex, but I think it's easy to argue why those might be relevant. So things like collagen, so mimicking that fibrous material of the ECM, and then other types of sugars, things like alginates and agarose that aren't found in the body, may be found in plants, which you've heard a lot about this week as well, but are easily tunable to change their material properties. And then I say hybrid. So again, our lab works a lot with this hyaluronic acid. So it's a polysaccharide found in the body, but we do a lot of chemical modification of it. So we retain some of those biological signals, but then also change it so that we can know and understand and manipulate the properties for our questions. And again, I already brought these up, but then how do we take each of these materials and then process it into those different types of structures, which I'll come back to. So polymer mechanics, so again, as a mechanobiology, when we think about these types of materials, we again can think about them as comprising both features of an elastic solid and a viscous liquid. So really all polymers have this behavior, and it's just the extent of which they have each of these types of properties. And there's a bunch of models out there to describe different types of materials. Some of these, like polyacrylamide, we consider them to be fairly elastic materials, whereas some of those viscoelastic materials have been designed to introduce some of that viscous behavior into it. And then we can test these materials a lot of different ways, and we'll get a different outcome based on how we test it, depending on the type of material that we're testing. So if we're stretching it under tension versus squishing it under compression, or applying some sort of shear force. And for different applications, things like tissue engineering, we might think about how that tissue is loaded in the body and then think about that as the way we test it. Here's just one example. This might be a tensile test of a polymer material. There's a lot of properties in here that you can get out of it. So ones that are really important, the slope of the stress versus strain curve can teach us about the modulus of the material. You might have a material that then yields, this is for polymers that maybe change their interactions as you continue to stretch it. They might have some maximum property here that might be the ultimate strength that you can obtain. And then at some point that material failed. So if I'm stretching it at some point, it's just gonna, that material is gonna break. And if you've done the test properly, it's gonna break in the middle so that you can really understand what is the stress that's applied or how much of that deformation or strain that leads to failure of the material. So we're interested in these really tough elastic materials in my lab in recent years. And so these materials will stretch a lot and deform and absorb a lot of load before they fail. And so that might be of interest, but other types of materials are very brittle. So if you just start to stretch it, you can apply a lot of load, but then it fails very quickly without deformation. So again, just I think keeping it, thinking about the different types of material properties that are important. This is early on is thought to be a linear response. This is based on the elastic nature. So you're just maybe changing the spacing of bonds, whereas once it goes nonlinear, usually you have slippage of polymers past each other. So again, you're changing that network. And if you release that load, it's not going to respond back. Whereas in the elastic early regime, it is. So yeah, that's a lot of quick mechanics. But mostly thinking about terminology, I guess. So you can get lots of information from the test and just depends on the materials of interest. A lot of mechanobiology, we're interested in things like atomic force microscopy. So this has a cantilever and some sort of indentation method. So this one has kind of a pointed one. A lot of times there's a bead on your cantilever. And you can go through and you can just kind of either look at the surface topography of a material, or you can test it by tapping on it and then measuring the force that it takes. So you'll see a lot of AFM used in mechanobiology because it gives you kind of a cell scale overview of the mechanics. Things like degradation, different mechanisms. So hydrolysis, this just means it has some chemistry in it that if there's water present, it'll start to break chemical bonds. Enzymatic would mean that if there's enzymes there, often produced by cells, or could just be added, we've designed these then to break those bonds in response to that enzyme. So a lot of these might have peptides that are known kind of interactors with those enzymes. And then some hydrogels just involve dissociation. So there's something that's holding them together into a network, and over time, they just kind of fall apart. So different mechanisms depending on the application. And there's lots of things to control the degradation behavior. So the chemistry that's used. So this is just a list of chemical groups like these polyanhydrides that can break down in the presence of water. So they hydrolyze. And the rate at which that happens is based on the bond that's used and the surrounding chemistry. So as bioengineers, we can tailor a lot of these properties. Things like hydrophobicity. So if it doesn't like water, so this on the right would be something that's more hydrophobic. So a drop of water beads up on the surface. That's not going to degrade as fast because it doesn't allow water present to break those bonds. Things like the molecular weight of the polymer defines how many bonds you have to break till it solubilizes. Things like the polymer structure. So if it's highly crystalline, getting things like water and enzymes into that material is harder. Things like the enzyme concentration, if it's enzymatic degradable material. And then surface area. So something that's really porous is going to degrade faster than something that's just a big, massive block of that same material because you can't get those water or those enzymes as easily into it. So this is one. So there's very common polymers that have been used in the biomaterials field. So these polyesters, maybe you've heard of things like polyglycolic acid, polylactic acid, or polycaprolactone. So this class of material, you can see it looks very similar because it has these ester bonds that will hydrolyze. But if I just put these other chemical groups around it, it'll change the rate at which this degrades. So as I go from top to bottom, this is more hydrophobic. So this polycaprolactone might take a year to degrade, whereas this polyglycolic acid might take weeks. And these have been used in a lot of things like sutures and biodegradable scaffolds and tissue engineering. And then what's nice is, again, we can synthesize these and we can then make copolymers of maybe that lactic and glycolic acid. And based on then the chemical composition, I can change the degradation. So we think a lot about degradation in my lab. So these handles that we can tune to change those properties. Then let me get through hydrogels and I'll pause, see if there's some questions before I talk more about applications. Again, I've already mentioned hydrogels, but these are water-swollen polymer networks. So you just think of these as made from polymers that really like water. So we can dissolve these polymers in water. And then if we link them together or cross-link them through different types of junctions, they won't dissolve then when they interact with water, but rather they'll just swell. So think about a three-dimensional kind of netting or mesh of these materials where they're linked together, but because, again, it likes water, those will interact with water really well, and so that whole material will swell. We can make them covalently cross-linked, that quite stable. We can make them through just ionic interactions, things like alginate. Those would be great for many applications too, and then eventually dissociate. Just physical entanglements can be used. So just these long polymer chains that are kind of wrapped around each other can lead to hydrogel formation. And then there's a lot of secondary forces like hydrogen bonding. Again, these are useful in mechanobiology because they can mimic things of tissue. So maybe they can mimic the elastic modulus of a tissue. These are really the only class of materials where we can embed cells within the material. So we can, if it's designed appropriately, put cells throughout the three-dimensional structure, and they're highly modular. So this is where I think the field's making now, altering how these polymer chains interact with each other to make them viscoelastic, changing the chemical nature of them to make them degradable. So I think these have had a long history of use in things like drug delivery, tissue engineering, and I think even growing more so in mechanobiology. We can make them a lot of different ways. So polyacrylamide gel, we just take a lot of monomers, so acrylamide, and then a crosslinker, a bisacrylamide. And with the right initiators, then these will form a hydrogel network. Our lab uses a lot of polymers like hyaluronic acid, where we just put chemical groups on it. So again, with the right crosslinkers or initiator, we can link those into a 3D structure. Again, most of these are going to be a nanoscale structure when we make them like this. And then just another example that was found in this kind of schematic is like an interpenetrating network, where we might take one network and then polymerize another polymer throughout it. And so you get some features or behaviors of both types of networks. So again, lots of modularity and ways that we can change properties. You can make them, often we make these in the hydrated state, and then they can either swell more or not. Or you can make them, some of these in the dry state, depending on the solubility of these different polymers and monomers, and then they can swell over time. But understanding their interaction with water is really important, so that drives the swelling. And this is important because it changes things like diffusion throughout the hydrogel, the surface properties, how cells interact might be based on the water content, of course, the optical properties and mechanics. And it's driven by a few things. So again, the compatibility of the polymer and the solvent. So again, that just means how much does that polymer, like water, will alter how it swells. And then this elastic retractive forces just means how much did I link those polymers together? So there's going to be some extent where it no longer swells unless it degrades or dissociates away. So again, swelling, I think, is important for a lot of applications. And it's driven by the thermodynamics, again, of these different features of the material. And really quickly, I just put on some different classes of hydrogels. I've already mentioned some of these. So covalently cross-linked would be very stable ones like the polyacrylamides, the polyethylene glycol. There's even within a PEG gel, there's lots of ways to make these. You can make these through a radical polymerization, a step growth reaction. The molecular weight of the PEG can be changed, the number of arms on a cross-linking PEG, and all this changes then what that network structure looks like. Again, physically cross-linked materials, one of these is polyvinyl alcohol that has a lot of hydrogen bonding in it. And some groups have used freeze-thaw methods that just drives the interactions between the polymers during that freezing and thawing. And so then that's those cross-linking junction. It's not through chemical cross-linking, it's just that physical interaction of those materials. And then a very popular one in the biomaterials field, alginate. So alginate, which is found in seaweed, and then just through different ions, in this case calcium, it'll just cross-link that alginate together into the 3D structure. And then kind of lastly here, stimuli responsive material. So we're really interested in this, and we have a few people in the lab really excited right now about materials that change based on some external stimuli. We're really interested in temperature responsive materials, but it could be the pH of that material, or it could be some sort of stimulation, light. I think that's probably temperature and light are the ones that we're most interested in. So how can we make a dynamic material? And maybe that's for drug delivery, so that dynamics releases a drug. Maybe it's for mechanobiology because that changes the material surfaces. And one of the most popular, I think, in the field is poly-NiPAM or polyanisopropyl acrylamide. This is a polymer that exhibits a lower critical solution temperature, or an LCST. That just means when I change the temperature, I alter things like the hydrophobicity of these polymers. So here, if you elevate the temperature, it collapses because there's more interactions between the polymers. And then it's reversible. So then if you cool it down, you get then more swelling. So these dynamic natures can kind of be imparted into materials. Okay, I'm going to pause there and just see if there's any questions on what I've talked about so far. And then I'll move into kind of those specific cell material interactions, and then you'll see examples in the field from that. But just curious if there's any questions yet.
Hi, Dr. Burdick. So I do cardiovascular research, and I was wondering, have you ever either run across or currently make like a good hydrogen model that like a 3D model that can like mimic like non-diseased and like a diseased artery? And yeah, whatever you recommend for that.
Yeah, that's a great question. You know, we've there's a fantastic lab down the hallway here in Boulder, Leslie Line one, who's really works on cardiac tissues and different types of diseases. So we've been making hydrogel models that, in that case, we just mix cells together with ECM molecules, so collagen, although others have done it with like fibrin, and we use the material and the way it's molded to then drive how cells maybe sense mechanics in their environment. And so then they remodel into these little tissues. And then in that case, they'll actually contract. And so this built some from work of Chris Chen's lab, Ken Margulies at Penn. But we've been exploring that a lot with Leslie's lab because she has a lot of iPSC cells with disease features to it. And so that's been a fun collaboration that I think is leveraging materials for those. Um, Vascular models, I think, I guess Bill probably talked about a few. We've done a little bit, but mostly for like microvasculature versus large ones where we can use 3D printing to make different kind of curvature that's found in vessels and understand maybe how that affects the cells. And then there's been a lot for kind of, large vessels. Usually a lot of those, like Bob Tranquilla, I think, has done some really nice work of like contractile gels where you put the cells and gel around like a post and then they just contract onto it. And so I find some of that work really fascinating. And probably what's common is all these are really relying more on the cells than the materials, but then giving a little bit of material design that might alter the features, such as the stiffness of that ECM that might then influence disease features. So hopefully that addresses or talks about a little bit. Of course, you could put cells on 2D surfaces of different stiffnesses and understand maybe that some disease feature, but I really like going to kind of the three-dimensional structure and context that those cells actually see during disease and trying to incorporate those into it. So
Yeah,
hopefully,
thank you.
hopefully that brings up some points of interest.
Thank you.
Any other questions? All right, I'll keep going along, and then hopefully at the end, I think the second half is really more. I shouldn't say more interesting because we really like the fundamental materials science, but I think it's where you actually see this then being used then in some of these questions. So I'll first just start a little bit about 2D. So one of the things is really trying to understand how cells interact with these materials in these different contexts. And I'm not sure if this has been brought up too much this week, but traction force microscopy is one method that our lab and many labs have used. It goes back to kind of these early papers that are now 25 years old, I guess. And in this case, cells are placed onto hydrogels that have beads that are embedded in them. And so often we try to do this in ways that the beads are mostly on the side that the cells are interacting with. And then you can watch these cells even over time and then monitor the displacement of these fluorescent beads. And then you maybe apply a detergent to then release that cell. And then the beads will kind of move back or relax into their original position. So this works really well with more elastic-like materials. Although I think there's been advances in many types of material systems, but this is a way that we can get then a map of the tractions that the cells are imparting onto that surface. So you can actually see how much they're pulling based on how much these beads are deformed. Here's just one example from one of our earlier studies looking at the cells on softer materials and stiffer materials. And so this got shifted a little bit. So this is on the stiff material on the top here, and then this is the soft one. And so you can see the rounded cell on the soft material has very low tractions, whereas this more spread cell on the stiffer material is imparting a lot of force then on that material. So that would have been correlated to larger bead deformations. And we've used this a number of interesting ways, I think, to look at over time. So if I take a material with a cell on it, and then I change the stiffness of that material, then I can watch how cells respond to that. And so it's taught us a lot about the kinetics of that cell transformation as it interfaces with two different types of materials. So over about I'd say maybe 4 or 5 hours is when we see them kind of reach that steady state, going from that soft to stiff environment. There's a lot of micropost systems. So Chris Chen's lab has looked at how cells sitting on these posts, if they're on very tall posts that can deform easier than on really short posts, they'll sense that environment, and then you can measure things like post deformation to understand those behaviors. So um, I'd be surprised if this wasn't brought up already this week, but these are some cool examples. So this is a cell, it's rounded because it's when it interacts through these posts that have some sort of ECM on them, you can see those just kind of bend really easily, whereas those exact same materials, same ECM ligands, but if they're really short, that cell can then sense that mechanics and then spread quite a bit more. These are mostly with elastomers, not hydrogels, whereas the TFM is mostly with hydrogels. There's been a lot of work. Again, this is kind of a paper now that's less than 20 years, but getting close. That's been around for a while that I think really started a lot of at least stem cell mechanobiology of how cells on surfaces or substrate of different mechanics behave differently, in this case related to cell differentiation. So this is with mesenchymal stromal cells and how they differentiate down different types of lineages. I think here's an example of some neural differentiation, some muscle differentiation, and then some osteogenic or bone-like differentiation as you change from very soft materials that might mimic that neural environment, intermediate materials that mimic that muscle environment, and then more stiff ones that mimic the osteoid environment within bone tissues. So this paper, I think, really kicked off a lot of mechanobiology and from Dennis Disher's lab at Penn. So again, I think Penn is, as far as mammalian cells, is really a center of a lot of mechanobiology. There's lots of ways to then also pattern cell interaction. So this is using an ECM stamping technique. So you can spatially control then where cells can interact with a material. And then this one was just depositing different ECM techniques. There's now many different ones. We use some other lithography methods in our lab. And here's just a few examples then of either cardiac myocytes on the top or neural cells. And by patterning ECM, we can really change how these cells organize on surfaces. So cardiac myocytes would be really disorganized if there wasn't patterning, but we can make this really elongated pattern that mimics then what maybe an adult heart looks like. And same with neural cells, they'll follow these paths. So you can look at things like neural connections between different cell types. So the technology, I think this field started with a lot of technology that's really advanced and now really available for applications. Using these stamping techniques, this is a nice example from Chris Chen's lab, again, very early work that then, again, opened up, I think, a utility of a lot of this. We've used this for some applications as well. They took those same MSCs and in a media that would support fat or bone-like differentiation, they showed that by putting cells on these islands of different shapes, if they're rounded like on the left side, in the exact same cells, same media, that shape would then dictate an fat-like differentiation. Whereas if you let those cells spread and then generate that kind of tension on the material substrate, those will form more like a bone-like cell. So I like this one a lot, and we did a little bit of this our lab as well, because everything's the same, and you're able to break it down to one question, which is cell shape. We can control topography. So common is putting just grooves onto these 2D substrates. This can change things like cell spreading. On the bottom here, the green is standing for focal adhesion. So the organization of focal adhesion changes. We can do this with fibrous networks as well. So they have a topography that's very different than a really flat material. So on the right here are just kind of some flat hydrogels. These are some engineered fiber networks. And you can see how different that cell looks than this one here that can spread almost too much, doesn't really mimic maybe what happens in the body. So all of these, so whether it's mechanics, topography through fibers, or this micro roughness, it's just guiding a difference of cell interaction that then can control downstream cell signaling. So whether that's differentiation or mimicking features of an activated cell that looks like they are in a disease state. This is where I think these substrates are pretty powerful. The same types of things can be done in 3D. I think 3D would, again, imagine a cell being surrounded by a hydrogel or maybe a fibrous network as well. It gets much more challenging, I think. How do you get the cells into that space? how do you monitor it? So of course you need things like confocal microscopy. So you're not just looking at cells in one dimension, you're actually looking and understanding what's happening in all dimensions. So I think it's tricky, but I think moving to this relevant state is really important. This is an example where that traction force microscopy that I mentioned for 2D has been transitioned into 3D. So in this case, the cell is placed into a hydrogel that has those same beads all throughout. This is a hydrogel that's been designed so that cells can degrade it to spread into it. So through some of those proteases. And then if you look at then, so again, you release the cell or kill it, so it loses those interactions. The gel relaxes back, and then you look at in 3D now at the differential or spatial change of those beads. You can then quantify that into bead displacement. And if you know a lot about the material properties, something like attraction force. So we've used this. This is an example from a paper where we looked at degradation of a material. So if a cell can degrade the material, it spreads a lot more into it. Whereas if it cannot degrade it, it just kind of retains as this rounded structure. And we showed that if you do that, then as they degrade it, they can then pull more on that material, which makes sense because they're spreading into it. And then using that same media, looking at fat and bone differentiation, if they can spread into it, they generate more attractions or bead displacement, and that leads to more osteogenic differentiation. If they're restricted, we've shown it doesn't matter what the material mechanics are. If their rounded shape is retained, then they can't generate those tractions. And so then that leads to a lot of fat-like differentiation. So this took a lot of kind of that 2D idea of cell shape and then imparted it into a 3D system. I mentioned viscoelasticity. So this is, I think about 10 years ago, this paper came out from Dave Mooney's lab, which kind of brought in, rather than just the modulus or the stiffness of a material, how it changes as a cell pulls on it. So this viscoelasticity, so again, bringing in that viscous component might change how a cell interacts. So in this case down here, these materials all have the same modulus, but as you go from left to right, you increase that stress relaxation or the viscoelasticity. And so that allows then these cells to spread into the material. So I think they really nicely showed that that's a parameter. Again, it's really hard to decouple all these features because of like adhesion might change in these materials or how they can cluster ligands to form focal adhesion. So you have to kind of keep track of that as well. And then 3D, again, important things would be things like porosity that again, I mentioned earlier. So a nanoscale porosity versus larger scale for foams and fibers. And that might change, again, how a cell interacts with it, how a cell can move through it. But all of these features that I won't go into too much detail again, but surface properties, mechanics, degradation, porosity, and pore size change based on how you process these in 3D. So here's just, again, hydrogels are nice because you can embed cells. So these are living cells that have been encapsulated into a 3D hydrogel. They can be injectable. So here's one that we had designed to be able to extrude through a syringe. So in our case, not only were the material properties important, but that we could actually inject it into cardiac tissue in this case. And then we can, again, tailor the mechanical properties. So that's how we bring the three-dimensional mechanobiology signals into this dimension with hydrogels. Bill mentioned this. I had this slide about incorporating these into microfluidics, which I think are really interesting. In this case, a study that looked at two-channel systems where you have vessels, and then you have another channel that has factors that'll diffuse to those vessels and then increase their cell migration. And in this case, looking at angiogenesis or the sprouting of new vessels. So that's a nice way to use these 3D hydrogels and processing techniques that had been described as far as microfluidics earlier. You can do things like photo patterning. So in this case, we looked at how cell behavior changed if we patterned in different types of crosslinks. And we did this with light. So in areas that we expose it to light, we just did this through some masks, we would restrict the cell spreading. In the other areas, the cell could actually degrade the material. And so now we could then look at differentiation, and we saw, again, that fat-like differentiation when we restrict spreading, and then that bone-like cell differentiation when we allow spreading. And so in one material, based simply on whether or not the crosslinks let the cell spread or not, we could get multi-lineage differentiation. Other things like photo degradation. So some groups like Christy Anseth here at Colorado and Cole DeForest at University of Washington have done great work to make a hydrogel. And then they can go in with light and then just degrade out regions because they respond to some specific wavelength of light to break chemical bonds. And so they've made some nice platforms with that. This is some work from that Cole DeForest lab looking at, rather than other ways to make channels, could they just degrade out channels with light? So they used a multiphoton microscope and could make these different lattices and then seed them with cells. So lots of ways to then bring that structure into 3D gels. So foams, there's lots of ways to make foams. You can use techniques like phase separation. So if you just, by controlling the polymers and how they interact, you can leave behind a porosity. You can put in particles, things like salt crystals, cross-link a material or let a solvent evaporate, and then leach out those salt crystals to leave behind pores. You can use gas processes to foam these structures to make, again, porous structures. So there's lots of ways to do that. These are typically very non-uniform pores, but I'd say fields like tissue engineering really started with things like these salt crystal foam processes. This is where I think 3D printing is making a big impact of how we can process these into three-dimensional structures. So different types of printing processes, like extrusion printing of these filaments, laser sintering of powder beds, inkjet printing where you just deposit droplets of materials, or light-based systems where you control through a liquid resin how you expose it to light. Our lab has really moved into a lot of these additive manufacturing 3D printing processes, especially since we moved to Colorado. So this is where we're exploring quite a bit of the technology right now. Additive manufacturing really just means you're depositing material to build up 3D structures by adding that material. So lots of types of materials can be processed. So hard materials from ceramics, polymers, and then of course hydrogels, as we've been talking about. And again, hydrogels being the one that we could put cells into three-dimensional materials. We often call this bioprinting. Some believe that you only call it bioprinting if you have cells involved during the printing process, which would only be possible with hydrogels. But we also think about bioprinting if we're making things that cells are intended to interact with, because then you have to consider the properties of that material and how those cells will interact with it. These are just some examples of extrusion printing. And so this one just builds up layer by layer from a surface. So this has an ink being printed from this nozzle. And then the printer just controls then the X, Y, and Z positioning. So a lot of groups will use these lattices because they have nice three-dimensional porosity to it. And then you can see here's just three-dimensional designs. So I think this has been used a lot, again, for tissue engineering probably, but if you understand mechanobiology, you can impart then into the materials in 3D printing features that could then guide cell behavior. These are just examples from a review paper of different types of hydrogels. So all these different natural materials, including things like gelatin and collagen, and then some synthetic examples down here, like those polyethylene glycol that have been processed using this extrusion 3D printing into 3D shapes. So now that the field has moved forward, we've got a lot of different kind of inks and materials that we can utilize. Just quickly mention a different type of printing that our lab spent a lot of time with, and we call this embedded printing or maybe suspension bath printing. It's got a lot of different names to it. This is the case that rather than extruding material onto a surface, you actually extrude it within the three-dimensional volume of another material. And sometimes then you use that material, then you wash away that material, so you're left with that 3D printed shape. Sometimes you keep that material and maybe wash out the ink to leave behind different channel systems. So really diverse ways to use this. And this, I think, is a technology that's been growing quite a bit. So we're actually organizing a special issue because now there's so many groups that are publishing on this embedded or suspension bath technique. But you can print really soft things, and there's no three-dimensional restriction then of where you deposit different types of ink. So this is fun. And then lastly, just really quick, fibers. So fibrous materials that might mimic some features of the extracellular matrix. Lots of technologies to do that. Again, phase separation. These kind of look like fibrous materials. Self-assembly of different materials that have maybe different domains that are hydrophobic and hydrophilic. These will self-assemble into nanostructures. And then a lot of technologies like electrospinning, where we can process polymers using the application of electrical fields to then form these little fibrous materials. This, again, is used a lot in 3D printing. Um, just looking at some of the structures and ways to make them. There's also techniques like melt electrowriting. It kind of combines 3D printing with electrospinning because you use an electric field to then define the extrusion of a melt polymer. And then the electric field helps you define it into these really precise structures. And then how you deposit this kind of filament of polymer, you can get all these different types of structures. And then there's been some work on weaving and knitting these types of materials into macroscopic 3D structure. But some of these are not very porous, so they might not be as useful for tissue engineering, but they're really strong. Whereas melt electrowriting is nice because you can make highly porous structures. And then I would think I just have a couple more slides, but electrospinning here, we've done this with different types of hydrogel materials. So you can make the same materials we've talked about with hydrogels into fibrous materials and look at how cells interact with those. And here's just some example of cells crossing a bunch of different fibers and looking at things like the density of the fibers, their alignment. So if you process these on something that's maybe rotating, you can collect them with a really defined or aligned pattern. And then I think I go back to some of these examples where if you compare these materials that are fibrous versus collagen versus flat hydrogels, again, the morphology I already showed you is very different. And these fiber networks mimic a lot more how cells interact with collagen, something that is a component of the ECM. And then other features, so because this is about mechanobiology, how cells then pull on that material. So one of the assays we use a lot with collagen is if a cell can pull on it and then degrade and remodel it, they'll take a solid piece of collagen gel and then contract it down. So you can see that here, and based a little bit on the concentration of the collagen. So these fibrous synthetic materials, they mimic that feature if you use the right properties, so they can contract it down, whereas most hydrogels, you never see that because the cells are locally interacting with that material, but it doesn't have the structure that allows it to interact across that entire material. Okay, so I think I'm going to end there with just summarizing what I've talked about applying materials in mechanobiology. So some of the take-home points, there's really a wide range of materials. So we talked about synthetic materials, natural materials, they're hybrid, and then processing them into gels, fibers, and foams. And this is where I think the biomaterials field, I mean, it's such a huge field because there's so many kind of things that we can do as a field to impart into materials, whether it's structure or chemistry, and that alters how cells sense and respond to it, and particularly related to mechanobiology. We can engineer properties and features to mimic the extracellular matrix. And so maybe deciding which features you want is one of the challenges. You can mimic things like disease by changing the mechanics or the cross-link density of the materials. We can guide differentiation based on maybe understanding the environment that the cells are in during development. Cells respond to various mechanics in both 2D and 3D. And I didn't emphasize this too much, but it can be very different in 2D and 3D. So the exact same material, if a cell sees it in 2D and can spread and interact with it, might guide the cell in a very different behavior than if they're in 3D in a confined environment where they can't sense it or interact with it the same way. So maybe then you need degradation to influence their ability to interact with those materials. So the context of this materials and dimensionality is really important. And then what we're really excited about for mechanobiology, for 3D printing is making highly complex materials that have properties like temporal changes in material mechanics or the patterning of different types of cell populations in 3D. And that's where techniques like, photo patterning, 3D printing, I think are emergent even more in defining our ability to ask those questions. So I think that's really where the field is going. So thank you very much for your attention. I'm happy to answer any questions that you might have.
I had a question. So I'm currently working on a microgel based platform, and I was wondering what kind of applications you guys are working on with that, because I've read a lot of your publications in that field. And then
Yeah.
also, I'm like starting to build on my project, so if you have any important like considerations or advice to include on that, that would be appreciated as well.
Yeah, thanks for that question. I didn't talk too much about microgels in this presentation, but it's probably one of the main classes of materials that the lab is using. And so I'll try not to be too lengthy, but in how I describe it. But this would be processing a lot of the hydrogels I mentioned today into microparticles. And so sometimes we call these microgels. And for those who are working on these, we can do that through just taking a gel and crushing it up, which sounds kind of wild, but that's actually used in a lot of clinical products right now in the field. So things like hemostats that are meant to absorb blood. But we often use things like microfluidics where you can make defined droplets and then cross-link them into microgels, or batch emulsions where you're just dripping in your microgel precursor solution to form these emulsions and then cross-linking it. So lots of ways to make them. We've used them for quite a few applications. I think the ones that I'll highlight, so then you take those microgels and then you kind of jam or pack them into these 3D structures. And what's great is you have some porosity in between the microgels, so you might harness that for some applications. But they behave almost as elastic solids, even in that jam state. So we've done quite a bit in 3D printing of these materials, showing that you can extrude then microgels as 3D printed inks. We're just submitting a paper now on also using that bath technology that I mentioned. So we can make a bath of microgels and we can print then microgels into that, cross-link it. And so then you can have two different types of properties or cells in a three-dimensional structure. And one of my postdocs, Kessie's, use this where he can make a temperature responsive microgel and a non-responsive one. So if you pattern them in different features, so imagine maybe on one side they're all temperature responsive where they collapse with light exposure, that can make things like bending objects because you can spatially pattern that. From a cellular perspective, so one thing we've been doing quite a bit in the lab is using these as injectables. So we inject them into tissues like the heart. So we've got an ongoing project where we're injecting these and then looking at cell invasion into cardiac tissue in vivo so that we can kind of understand then how we can design them to promote then the repair of that heart tissue. And then from an in vitro standing, probably one of the things we're most excited about right now is we've been taking aggregates of cells, so cell spheroids, mixing them with microgels. And one of my students, Nico, published a paper on this just a few months ago in Advanced Materials. And so then if you get contact between the microgels and contact between the spheroids, you can grow this into a three-dimensional tissue by how spheroids interact with each other and use the microgels to stabilize it. So if we just put a bunch of spheroids together, they just form a big ball that probably cells die in the middle. If we use these microgels, we can control the shape of what it becomes through that structure and then let them grow into a 3D tissue that then retains that shape. So yeah, we're using it for a lot of different things, and I won't go through other parts of it, but yeah, drug delivery vehicles, tissue engineering. The second part of your question, I think, was like suggestions or guidance on it. So probably maybe some challenges are, I think each student develops their way to like make the particles and then pack them in. So just making sure you're developing reproducible protocols, but then also harnessing the reasons you want to use microgels, this diversity in, you can take 10 populations of microgels, mix them together and get this highly complex material that you'd never get with just a uniform hydrogel. So I think exploiting the advantages of microgels is one of the highlights. So please reach out if you want to talk more about microgels. As you can tell, I'll talk forever about microgels. So thanks.
Thank you. I'm sure I'll have more questions for you.
Yeah, please do.
I also had a question, not about the micro, but on one of your pictures when you were going over the fibrous networks, I saw that you were showing like right there on that one, you have like vinculin, actin, and like the nucleus. I was just curious if you knew why like the vinculin was like super close to the nucleus and not more like at the endpoints of the actin.
That's a good question. Yeah, I don't know. Yeah, a lot of the 2D ones, and maybe this is like a cell that's kind of migrating around. A lot of it we'll see the more of the yeah, those punctate focal adhesions. As you can see, this I think is a beautiful image actually, because it really shows the actin structures really nicely. And I think that's because this cell just flattens out so much into like a little pancake. And you can kind of pinpoint those. Some of it might be imaging artifact, how it's hard to get maybe the visualization of some of these things in this context. You'll see here in some of them the vinculin. So there's probably actin throughout this whole kind of cell, but it's so hard because it's kind of compacted to see it. And we might have just found a cell where this is where there's been one fiber. I don't label the fibers here, which might help explain some of that. Like where they're really attached and interacting with the fiber is probably going to define where those focal adhesions are. So I think we'd probably see a lot of different behaviors, and this just happens to be how that one cell is. But yeah, we didn't look probably as specific into that as we could have. And this one's probably a soft one too, so it's clustering these fibers. And so where those fibers are and how it's being presented to the cell is really going to change, I think, that organization of those. So yeah, thanks for that question. I don't think we looked at that specifically.
Thank you.